For this video, I'm going to cover A2 topic 18 on classification, and I'm going to focus on 18.2b, where I'm going to focus on this learning outcome because it's actually new in the sense that the domains are included only after 2016 syllabus. So there might not be many questions on this, but let's get prepared. Before I go into the details of three domains, do you know how species are being classified? So the first level is actually the domain, where we actually have three domains, which is further split into 5 kingdoms, which is what we're going to learn, and followed by phylum, class, order, family, genus, and species. So usually if you come across the word like Vibrio cholerae, the Vibrio will be genus and the cholerae will be species. Another example would be like Homo sapiens. Homo will be the genus and species will be the sapiens. So now we're going to go into the details of three domains. Classification of these three domains actually was discovered recently when scientists realized that under the prokaryotic kingdom, it's not as simple as you think, as in the bacteria, they are actually branched into 2 different categories. And they realized that this Archaea is actually quite similar to the bacteria, but yet they have some of the features that are related to eukaryotes. So now we're going to look into the detail about a bacteria and see how an archaea is different from bacteria, therefore they can't be categorized under bacteria, and finally the features of eukaryotes that we are familiar with. Now I'm going to go through with you the essay points for each of the domain, and then from here we're going to extract the details and see how to state the differences. So first one, when you look at this essay, who do you think it's describing about? DNA is not surrounded by nuclear membrane. So this is where we know that it sounds like a prokaryote. Let's look into detail. The DNA is circular without histone protein. Here the essay, they accept naked DNA, but as we know that the naked basically means that it's without the histone protein. Do you know how to differentiate between a circular DNA and plasmid? Okay, let me show you. Recap on the structure of a prokaryote, the bacteria. So bacteria usually has a cell wall, and the cell wall is made up of peptidoglycan, or some of you might be more familiar with murine. And the bacteria would have a circular DNA, which doesn't really look circular because they are randomly coiled. And plasmid is usually a very small circle DNA, which not necessarily all bacteria has it, but if a bacteria has it, it most likely consists of an antibiotic resistant gene, therefore giving resistance to antibiotic. And other than that, they do not have any double membrane organelles. Examples like no mitochondria, no chloroplast, and no nucleus. No single membrane bound organelles such as ER and Golgi. But they do have ribosomes. And the ribosomes are actually the smaller ones, known as 70S ribosome. And if you know the size, it will be known as 18 nanometer. Some of the bacteria has an external layer known as the capsule. And some do have a flagella for movement. Bacteria usually exist as a single-cell organism known as unicellular. So this is where you can make use of your AS knowledge. So you have less thing to memorize because you have already memorized it. Moving on to this essay, you will see that the description is similar to a prokaryote, but let's spot something different. No nucleus, circular DNA. Seems familiar, it looks the same like bacteria. That's where we need to identify points that are different, such as the membrane composition is different from bacterial eukarya. So that means it's not made up of phospholipid. No membrane-bound organelles, 70S ribosome. Cell wall present but not made up of peptidoglycan. So now we know that this is not talking about a bacteria, but instead is known as Archaea. So what else makes Archaea different from bacteria? This Archaea actually has a histone protein associated with DNA. So sounds like a eukaryotic cell. And some of them are extremophiles, whereby they live in extreme environment such as volcano vent, a hot spring, or anaerobic condition. And finally, I think this one most of you are familiar. It contains a nucleus, and these cells contains membrane-bound organelles, ATS ribosomes, and they had chloroplast mitochondria, linear DNA with histone protein, and they could be existing as a unicellular or multicellular, divide by mitosis, and can be sexual and asexual. This is definitely a eukaryotic cell, where they belong to eukarya domain. Let's summarize the difference between the three domains. And we're going to start with the bacteria and eukarya, which is something that we are more familiar with. So ribosomes. Bacteria actually have 70S ribosomes, whereas eukarya actually have both 70S and ATS, but we're going to focus on the difference. Therefore, we're going to write ATS. Membrane component, bacteria is made up of phospholipid. Take note, it's a membrane but not cell wall. And eukaryote also the same thing. We have phospholipid bilayer. For the cell wall, bacteria is made up of peptidoglycan, or known as murine, whereas eukaryotes is actually made up of cellulose for plants and chitin for fungi. As for DNA, bacteria is circular without histone protein, and they are suspended free in cytoplasm, as a bacteria do not have a nucleus. So compared to eukarya, the DNA is linear and is coiled into a chromosome as it's associated with histone protein and is contained within the nucleus. Membrane-bound organelles for bacteria, absent, and for eukarya you have double-bound membrane organelles such as mitochondria, chloroplast, ER, and Golgi. As for other features, we can mention that bacterial cells are actually unicellular and they reproduce by binary fission. Whereas for eukaryotic cells, we are usually multicellular, but some of them can be unicellular. And our cells can reproduce by mitosis sexually and asexually. Examples of bacteria will be something that we have learned in AS, Vibrio, cholerae, and for eukaryote will be including the 4 kingdoms such as fungi, animal kingdom, plant kingdom, and protoctista. So let's look at how Archaea is actually different. Archaea is known as CHAEA. Okay, ribosome. You can take it as 70S, or some they will mention that it's even smaller. Membrane component, actually they are made up of different composition, so we are not expected to know that. We just need to know that it is not phospholipid. Cell wall, not peptidoglycan or not murine. As for DNA, it is still circular, but is known as a circular chromosome as it is associated with histone protein. And it's different in this way, but yet it's suspended in cytoplasm as they have no nucleus, no membrane-bound organelles, and it's also unicellular and divide by binary fission. So examples will be extremophiles. Sometimes we call it thermophiles, where the bacteria actually stays in extreme high temperature. Hope this table gives you a clearer idea on what's the similarity and differences between the 3 domains. For more understanding on the other sections of this topic, do not hesitate to contact me for group or private tuition. Thanks for watching.
That's all for today's video. If you're interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with 1 or 2 past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.